So good afternoon, everybody, and welcome to the BioFoundation lecture series. It's a real pleasure today to be able to introduce today's speaker, so Dr. Nicolai Franzmeier from LMU Munich. He was this year's Early Excellence in Science Award for Medicine. I'd just like to take a moment to introduce the BioFoundation and to announce the launch of a really exciting project for us, and I think really of interest to all of you. So I think many of you already know this is the first year we've built a connection between the BioFoundation and the Bio R&D Research Organization by hosting 6 webinars. This is the 5th so far, bringing our prize winners to you, the R&D community. We have one more this year that will be from Stefano Barco, who was the winner of this year's Biothrombosis prize. So the foundation is legally independent, as you know, from Bayer AG, but we do have the same vision to achieve health for all and hunger for none. As we strive towards this, we focus our work in 3 key areas. The first is equality in science. Now, depending on your environment, you might instinctively think of gender equality. We talk a lot about global equality in scientific capacity, and that's actually what I'll tell you a little bit about today on the next slide. But we also talk a lot about the role of society in science, and that's also very important for us to really move forward and provide solutions that are relevant for a global population. Collaboration in science, something you're all very comfortable with. We know that interdisciplinary research and innovation really happens at the interfaces of disciplines, and only by sort of working in these complex environments are we able to address the complex challenges we have in health and nutrition. Though again, I'd like to mention the role of society in science, especially as we go towards our 3rd key area of work, and that's looking at trust in science and technology. If society does not trust, does not engage with new innovations, new technologies, unfortunately, they cannot be implemented, and we do not have the impact we wish to have with the really exciting science that goes on across the world. So I just take a moment to tell you about a brand new project that is coming to fruition. So the research hub that we sponsored together with the Alexander von Humboldt Foundation will be launched formally in January next year. So we are supporting, you can see on the right hand side here, Professor Daniel Olusolo Ojoongbi. He's a medical macrobiologist at Ladoke Akintola University in Nigeria. And with funding of €750,000 over the next 5 years, we're supporting the establishment of a Center for Emerging and Re-emerging Infectious Disease in Nigeria with a partner site in Sudan. Now, their main research topic is looking actually at the potential genetic basis for the reduced mortality and morbidity as a consequence of COVID-19 infection across the continent. That I have to say that's just the first step in a broader research program. One of the reasons we love this particular hub so much is really their commitment to not only the next generation of scientists. So there are 3 postdocs, 5 PhDs just in Africa funded through this, more in Europe. But they also go a step beyond to educate the broader scientific community in their countries through bioinformatics training, best clinical practices for inter-epidemic time periods, and long-term, we will establish a biobank in Nigeria and Sudan for continual studies within their areas of research. So we're very proud to support that over the next few years, and more information will certainly come. So with that, I would like to begin by introducing today's speaker, so Dr. Nico Franzmeier. So Nico, please feel free to start sharing your slides and turn on your camera. There will be just to mention, we posted a few links in the chat if you would like more information on our collaboration with Alexander von Humboldt or the BioFoundation in general. There will be a Q&A at the end of today's session, so please use the Q&A function. I will moderate this, and should you wish to join us after today's talk for actually an open discussion with Dr. Franzmeier, you should have already received a link from us if you are a Bayer employee. If you haven't received this and would like to join, please just drop your email address in the chat and we can send you that directly. Okay, so Nico, it's a real pleasure to have you here today. Dr. Franzmeier actually began his career in Austria at Innsbruck University studying Physiology and Medicine. He then moved to the LMU hospital in Munich to study Systemic Neurosciences and graduated his PhD in 2017. And he then stayed there to undertake first postdoctoral research, and then this year he became junior group leader within the Institute of Stroke and Dementia Research. Dr. Franzmeier's main topic or main focus is Alzheimer's disease neuroimaging, and he combines structural and functional MRI and molecular positron emission tomography, so PET imaging and genetics. In 2017, he won the Steinberg-Krupp Alzheimer Research Award and is actively engaged with the Alzheimer's Association, co-chairing more than 1 or 2 specialist groups there. So Dr. Franzmeier, Nico, we're really happy to have you here today and really looking forward to your presentation.
Yeah, thank you, Karl, for the very nice introduction, and I hope you can all hear me well. So yeah, it's my real pleasure to introduce some of the work we're doing today. And as Karl already mentioned, my work focuses mainly on Alzheimer's disease. And I would like to give a brief introduction on how we currently understand Alzheimer's disease, which is best understood as a cascade of pathological brain changes. So if we look at an axis of time, we can see that one of the first brain changes to occur in Alzheimer's disease is assumed to be the brain-wide accumulation of a pathological protein called amyloid beta, and this accumulates in a brain-wide manner up to 20 years before patients actually develop the first symptoms. And this development of amyloid beta plaque pathology is then, after a certain time, followed by the spreading of the second hit, so to say, which is tau pathology, which spreads from the inferior temporal lobe, shown here, throughout the brain. And once these pathologies have both accumulated, we see neurodegeneration, meaning that neurons start to die. We see brain atrophy on MRI imaging. And ultimately, these brain changes in conjunction lead to the development of cognitive impairment. So something that we call MCI, or mild cognitive impairment in the clinic, up to the level of dementia. And our lab in Munich has 2 primary research interests. First of all, to understand why does and how does the primary pathology of Alzheimer's disease progress throughout the brain? And second of all, how does the pathology impact cognitive function? And here we put a particular emphasis on understanding why there is such a large variety in the cognitive ability of individuals at a given level of brain pathology, suggesting that some individuals actually have high reserve or resilience. That means they can better cope with brain pathology. And I'll introduce that in a minute. And to study these major questions, we combine, as Karl already mentioned, PET imaging and biofluid biomarker data together with connectomics using mostly fMRI and to a certain degree, and I'll just briefly touch up on this topic, genetics. And I'd like to start with introducing some of the work that we did. This was actually most of my PhD work on reserve and resilience, or how we believe that people can manage or cope pathology. And there has been a lot of demographic and epidemiological evidence showing that people with higher education, people with a more active lifestyle that are physically active, or people which are more intelligent, smarter, show an attenuated effect of brain pathology on their level of cognitive decline. And this is not only true for Alzheimer's disease, but for basically every brain pathology that you can imagine, like frontotemporal lobe or dementia, but also stroke. But one substantial question that has been unaddressed for several decades now is how do people with higher education, activity, and so on actually manage to better cope with brain pathology? So what is the substrate in the brain of reserve? And to study this, we are using resting state functional MRI imaging. So this is a technique where we can observe how brain regions interact from a functional perspective and how they are connected to each other. And using this technique, we can derive different functional brain networks. So here you see superimposed on that brain surface what we call the cognitive control network, and where these red regions are interconnected to each other, so they communicate. And this particular network has been shown to be critical for higher level cognition, so for top-down attention, for cognitive flexibility, and so on. And we became particularly interested in this network in the context of reserve and resilience, since previous studies could show that higher connectivity, so the more this network was interconnected, was associated with higher intelligence in young individuals. And in a preliminary study in patients with Alzheimer's disease, we also found a slight association between individuals' education levels and the connectivity of the control network. So we became interested to ask whether the control network actually supports reserve in Alzheimer's disease. And to this, this was one of the first studies that we did in 2017. We combined resting state fMRI imaging together with glucose PET to measure the neuronal damage that occurs in Alzheimer's disease patients' brains. And in this precuneus brain region here, typically patients develop a glucose hypometabolism, so neural damage, which goes hand in hand, as you see here in this red scatter plot, or red part of the scatter plot, with an impairment in memory. However, we found this only to be the case in individuals who had relatively low levels of control network connectivity, whereas individuals with high control network connectivity, this association was nearly absent. And that was the first indication for us to see that the control network may in fact support reserve in AD. And then we went on to validate or replicate this finding in a more clearly phenotyped cohort of individuals, so patients who have inherited a mutation which determines them to develop Alzheimer's disease. So these are very rare cases. And in these individuals, you can track the abnormality of brain changes throughout the entire disease course, so 20 years before symptom onset up to 10 years afterwards. And the symptom onset is estimated in individuals based on the age when their parent from which they inherited that mutation actually became demented. And in this cohort, we can see that amyloid PET levels in green increase over time, the level of abnormal tau pathology increases over time, and we observe neurodegeneration in terms of glucose metabolic change and atrophy of the hippocampus, so a brain region that is key for memory. But when we superimpose the cognitive trajectories for individuals with low versus high integrity of their control network, we see that the dashed line representing the individuals with more higher integrity of their control network actually show a shift of their cognitive trajectories towards a later time point, suggesting a certain buffering effect on the impact of brain pathology and cognition. But this doesn't tell us by which brain mechanism the control network actually may support reserve. And we know from cognitive neuroscience that the control network, shown here in yellowish, supports a segregated and modular brain architecture, which actually facilitates cognitive performance. So we were wondering whether a more segregated and efficient brain network topology is actually the driving force between that renders people more resilient. And we checked this again in two independent patient cohorts, one which I've just shown with inherited Alzheimer's disease, and another cohort with sporadic Alzheimer's disease. And here you can see that people with more segregated brain networks, so more modular brain architecture, show an attenuated cognitive impairment over the disease course. And very similar, so in sporadic AD, we found that the higher the tau levels, typically more abnormal cognition becomes. So this is a measure of global cognitive performance. However, this effect was also attenuated in individuals with high network segregation. And with this, we believe that we have tried to address the long-going or long-standing question why protective factors have a buffering effect on the association between brain pathology and cognition, and that part of this may be explained by individuals' brain network topology that then renders individuals' brains more efficient. And with this, I would like to conclude this chapter on reserve and resilience and focus on what has become one of our major interests in the lab during the last couple of years, namely the understanding of how does primary AD pathology actually develop and progress in the brains. And just because of a timing perspective of what brain change occurs first, one line of research focuses on amyloid pathology. And we know that when amyloid accumulates in patients' brains, that this triggers a reactive inflammation. So microglia, the brain resident immune cells, become activated. And this has been known for quite a long time. So here you can see, for example, a microscopic image of an amyloid plaque shown here in blue and surrounded by microglia shown in white. And they really encapsulate amyloid plaques, shield them from the surrounding, and also to a certain degree phagocyte these amyloid deposits. And part of this microglial response to developing amyloid pathology is mediated by a surface molecule called triggering receptor, or TREM2, which is shedded and which we can in fact measure in the cerebrospinal fluid of patients. So when we do a spinal tap and extract cerebrospinal fluid, we can go to the lab and actually measure levels of soluble TREM2, which are a proxy of how activated microglia are in patients' brains. And my former colleague Mark, who is now in Barcelona, has tracked the levels of TREM2 so microglial activation over the course of autosomal dominant and sporadic Alzheimer's disease. And you could see that there is a reactive increase in brain inflammation levels over time, which then vanishes once people become more and more severely ill. But this nonlinear trajectory was very well replicated across these different disease models. And one question that we had very early was whether a TREM2 related microglial response is something that is protective for the brain, or whether it's detrimental and actually contributing to the further progression of the disease. So to first test this, we went to a mouse model. So transgenic amyloid mice who carry a couple of mutations that render them prone to accumulate amyloid. And we combined this amyloid PET imaging in those mice with TSPO PET imaging, which is an imaging modality with which we can visualize the microglial activation in living animals or patients, if you will. And we found that the higher the microglial activation at baseline, the slower the subsequent rate in amyloid accumulation was in this animal model, suggesting that actually inflammation may not be such a bad thing, but actually help those animals here to maintain brain integrity longer. But we then went on to validate this finding in a translational manner in patients with Alzheimer's disease, where we combined longitudinal tau PET imaging to track the accumulation of amyloid over time with cerebrospinal fluid measures of TREM2. And here you can see that over time, which is plotted on the y-axis, the amyloid accumulation rate, people typically accumulate amyloid while being at intermediate levels of their global amyloid deposition. However, you see that this accumulation rate in amyloid is attenuated in the yellow group, so in those individuals who have higher microglia activation compared to those with lower TREM2 levels. So again, here we have an indication that both higher TREM2 and TSPO PET in a translational manner are associated with reduced accumulation of beta amyloid in the brain. We then went out to ask, does that actually translate into clinical benefit? So is that just a biological phenomenon that we observe, which doesn't manifest in any clinical consequence? So we looked at a study group that was tracked for roughly 10 years over time where we had TREM2 measures at baseline and cognitive performance at baseline over time. And we found in this group of patients that individuals with higher TREM2 levels in blue showed less of a decline in cognition over time compared to the group with low TREM2 levels. And in a survival plot, you can also see that the conversion rate to mild cognitive impairment or to dementia is actually reduced in these individuals, together suggesting that we have in these clinical cohorts, so these were all cognitively impaired already, we see a benefit of microglial activation on cognitive trajectories. And then we went on to ask, does that generalize to individuals who are just at risk of AD? So people who carry a genetic risk factor, the most common, which around 20% of the population has, which is called ApoE4, which is associated with a, I think, 4 times higher risk to develop Alzheimer's disease. And in these individuals, we looked again stratified by the levels of TREM2 at the rate of cognitive decline over time. And the more positive the rate of change, the more declining are people. And we see again that people who are at risk of developing AD with high TREM2 show basically an attenuation of their cognitive decrease over time. So that in this at-risk, non-clinically impaired yet population, and at the level of neurodegeneration, so when we look at hippocampal atrophy over time, we actually see the very same thing, that people with low TREM2 levels show faster atrophy over time compared to people with high atrophy, together indicating that a certain level of reactive brain inflammation may in fact be something that is protective, and this is currently explored in several, I think already phase 2 clinical trials to see whether targeting inflammation may actually be a therapeutic strategy. And with this, I'd like to conclude the chapter on amyloid and focus on the second and, to my, as I believe, most critical brain pathology that develops in Alzheimer's disease, which is tau. And just to briefly clarify what tau is, so tau is, in contrast to amyloid, an intracellular pathology. So tau is typically located attached to the microtubules, and within the context of Alzheimer's disease, tau detaches from these microtubules, and from those fibrils and tangles, and the axon degenerates, microtubules disintegrate, and so on. And what has been a very important discovery already, I think 20 or 25 years ago, was that tau has a prion-like spreading pattern. So when tau spreads across synaptic connections, so when there is a tau harboring neuron, these pathological tau fibrils can actually be transmitted across the synapse, infect the downstream neuron, and they induce template-based misfolding of physiological tau. So we have a cascade-like, infection-like propagation of tau. And the spreading of tau pathology is really one of the key drivers of cognitive impairment. I mentioned initially that people develop amyloid up to 20 years before they develop symptoms. With tau, this is quite different because tau occurs really shortly before people become cognitively impaired. So Heiko Braque and his wife earlier, 1991 already, described this Braque staging pattern of pathology, suggesting this gradual increase of tau over time, which could for a long time only be tracked in post-mortem data. But with the development of tau PET imaging, where we inject a radiopharmacological tracer that can visualize tau in living individuals, we can now do this staging already in living patients and stage how far along the road in terms of Braque staging are patients already. And this is work that has been done by Davina, postdoc in my lab, who looked at the level of Braque stage across individuals and the cognitive decline over time. And what she found was that the further people were down the road on their Braque staging, so the more widespread tau pathology was in the brain, the faster the cognitive decline was over time, together suggesting that the development of tau is really critical for developing cognitive impairment. And to better understand how tau develops and how tau spreads throughout the brain, we aim to translate this transsynaptic tau spreading model and the postmortem-based tau spreading scheme to the in vivo level to really study these mechanisms in living individuals, or at least a proxy of it. And to this end, we combine, again, functional connectomics to study the connection between brain regions together with tau PET imaging. And in two of our first studies, we combined longitudinal tau PET imaging. So we tracked individuals on tau PET over roughly 2 years with resting state fMRI connectivity data. And we here use the brain atlas that parcellates the entire cortical surface into 400 smaller regions. And then we can look at how are these cortical regions interconnected to each other, giving us such a connectivity matrix where more bluish colors mean these two regions are more interconnected to each other. And very similarly, we can obtain the covariance in tau accumulation between regions. So how similar is the rate of tau accumulation over time between two different brain regions? And when we looked at the association between these two measures, we found that brain regions who had a higher likelihood of being connected to each other also had a more similar rate of tau accumulation over time, which gave us a first indication that the connectomic network architecture of the brain may give us some information about how tau spreads and tau accumulates in the brain. And we then went on to a second sample to replicate this finding. So the biofinder cohort provided by our colleagues from Sweden, where we found the very same association, suggesting that higher connectivity is associated with more similar tau change across regions. So now this scatter plot here looks quite like a large cloud. So we split this further up by brain networks to see whether this is driven by any particular brain network that might be vulnerable to tau pathology, but this was not the case. So when you look at these colors, which represent the brain networks shown here on the surface on the left, you see that this association between connectivity and a higher likely or higher similarity in tau accumulation was evident across the entire brain, suggesting that we have a ubiquitous phenomenon. And based on this finding, we then asked, can we actually use the connectomic roadmap of the brain to forecast how tau will spread in a given individual? And to this end, we again made use of two independent data sets which covered the entire Alzheimer's disease spectrum. So we cover people from being cognitively unimpaired or cognitively normal, that's why CN, but with elevated amyloid pathology in their brains, towards mild cognitive impairment and people with AD dementia. And the colors here indicate the level of tau deposition obtained via PET. And you can see that over with increasing disease severity, tau levels become stronger. And by obtaining data across the entire AD spectrum, we can estimate a spreading sequence of tau using such a hierarchical matrix approach, where we see that brain regions here in bright show early tau abnormality, and brain regions in dark purple show very late tau accumulation across the disease trajectory. So basically, it allows us to show across the population regions which show early tau and regions which show late tau. And this also allows us to determine those brain regions in which tau most likely starts, brain regions which we call the epicenters of where the catastrophe begins, if you will, in which tau emerges first and from there takes its route throughout the brain. So we took the topological information of these epicenters, which are in line with the findings by Heiko Brag in the inferior temporal lobe, and mapped how are these brain regions connected to the rest of the brain. So we took fMRI again, mapped the seed-based connectivity pattern, and we found that the connectivity pattern was highly informative about the sequence in which tau propagates throughout the brain, such that brain regions which were closely connected to an epicenter, here shown on the left, showed tau abnormality very early on in the disease course, whereas brain regions who are here on the right show tau very late in the disease course. So by knowing the connectomic architecture of the brain, we can make a prediction about how tau propagates throughout the brain. But this here clearly has a downside, since you can here see that this is not a very clean separation. So we have large heterogeneity of tau deposition across individuals. And this just reflects clinical reality, because not every patient is the same, not every patient manifests with memory impairment, but people can be very different in terms of their clinical phenotype, depending on the pattern of tau pathology that they have in their brains. So we try to make use of this large heterogeneity to test whether our connectomic models can actually predict this heterogeneity in tau spreading. So we decomposed the tau PET imaging data set using an independent component analysis, so pattern separation approach, into 9 different subgroups with heterogeneous tau deposition, and more reddish colors here mean more abnormal tau, and each column is one of these subgroups. And you can immediately see that there is very large heterogeneity. So we can see, for example, one subtype in which we have tau pathology more or less only in one hemisphere, in the occipitotemporal lobe. And then again, we have other phenotypes which develop tau completely elsewhere in the more neocortical regions, so in the parietal lobe. And then we applied our connectomic spreading models to these subtypes and tried to test whether also in these heterogeneous individual or subtypes, tau spreading follows the connectomic roadmap of the brain. So we applied a sliding window approach here just to give an estimate of how does tau propagate throughout the brain. So we see very early on. So if you believe this could be like an early time frame, this is a late time frame. And early on, there's not much tau accumulation happening. But at some time point, we see in this subcluster that tau starts to develop in the occipital cortex, and then from there it makes its route throughout the brain. And when we then again apply our hierarchical modeling, we identify the epicenters here in one hemisphere in the temporal and occipital lobe. And we can again see that this spreading sequence that you observe up here clearly follows the connectivity pattern of those brain regions that you see here. And just applying this very same analysis scheme for a quite substantially different phenotype, where we observe tau pathology first in the parietal lobe, from where it then travels to the rest of the brain, we can again define the epicenter, so the regions of earliest tau PET here in the parietal lobe. And we see that when we look at how are these regions connected to the rest of the brain, this gives us a good prediction about which brain regions develop tau next. So this was all group level data. And we then went to the patient level. So in terms of a precision medicine approach, trying to define for each individual when he comes to the clinic or she comes to the clinic, a tau epicenter at baseline. So we look at the tau PET scans, take the brain region which has highest tau, and we assume that those brain regions which are most closely connected to a given patient's epicenter should show the fastest accumulation of tau over time, whereas regions which are far away in terms of connectivity should show late tau accumulation, indicating this gradual spreading pattern here from the temporal lobe throughout these connected regions. And we tested this in 3 independent samples, all with longitudinal tau PET data, all amyloid positive, so all Alzheimer's disease cases. And we looked at longitudinal accumulation of tau here on the y-axis and stratified the accumulation by these quartiles that were defined by connectivity. So Q1 is a brain region that is determined for a given individual that is really closely connected to a given patient's epicenter. And across all 3 cohorts, we see that actually with this zooming in approach of predicting where will tau accumulate, we can actually quite sensitively measure where will tau accumulate over time. And this is clinically meaningful in times of surrogate endpoints in clinical trials, where we need a biomarker or where we determine a biomarker change or a reduction in biomarker change as a treatment of being successful or not. Because with this measure of patient-tailored endpoints, if you will, we outperform conventional metrics of obtaining tau accumulation over time, such as Braak staging or basically looking at the entire brain. And we then simulated clinical trials suggesting different endpoints like Braak staging endpoints, whole brain regions of interest, versus the patient-tailored method that we're predicting. And we found that just projected numbers needed for a clinical trial in order to determine intervention effects of 20 to 40%, we have actually the highest power to detect such an intervention effect. And this is currently implemented exploratorily by Avid Radiopharmaceuticals in the US. So this topic was all about predicting the pattern of tau accumulation, but can we also use connectivity to predict the rate of tau accumulation? So how fast does it actually accumulate? And to this end, we came up with the hypothesis that basically the brain network, the brain can be parcellated into globally connected hub regions shown in red. So brain regions who show a lot of connectivity to their periphery. So these would be like in roadmap, a city like Munich that has a lot of roads connecting different other cities, whereas other regions are more peripheral and do not show so many connections. And we reasoned that if tau pathology hit such a hub early on in the disease course, then tau could have many ways to go. So speaking of infectious measures here, we would have a high R value if tau occurs in such a hub region early on, whereas if it occurs in such a peripheral region, we would find rather little tau spread. So Lucas, a former MD student in the lab, took these individual tau PET patterns of patients and assigned them to a gradient of whether an individual's tau PET pattern was falling in non-hub regions, so regions which show very little connectivity, versus patients who showed tau in hub regions, so where tau really targeted these densely connected brain regions. And what we found again across two independent cohorts was that the more tau pathology was biased towards these globally connected hubs, the faster the accumulation rate over time. So with this measure here, we can make a prediction about how fast will tau accumulate in the future. And in a complementary approach, we try to also understand what are factors that modulate the accumulation and spreading of tau pathology. And here we've recently started to look at genetics. And one of the key genetic risk factors in the BIN1, so bridging integrator 1 gene, which has been previously by preclinical data related to tau pathology, in particular to the endocytotic uptake of tau pathology, so pathway which is hijacked to enter neurons while during this transneuronal spreading scheme. And we just stratified a group which underwent tau PET by carriage of this BIN1 risk allele versus the normal allele. And throughout the entire brain, basically, we found that these risk allele carriers showed higher tau pathology, giving for the first time in vivo evidence that this risk factor is actually related to tau pathology. And when we looked at the same association using amyloid PET imaging, we didn't find such an increase. And in a follow-up study, we then asked whether this translates into faster tau accumulation. So we believe that amyloid triggers tau, and that this amyloid-related tau accumulation may be facilitated in people who carry this risk allele because their transsynaptic spreading mechanism may be increased. And this is in fact what we found. So we found that people with higher amyloid PET and carrying the risk allele shown here in yellow showed faster accumulation of tau over time compared to people who did not carry that risk allele, again consistent across two independent samples. And with this, I would like to leave the Alzheimer's disease field and move over to another class of neurodegenerative diseases in which we recently got interested in, because tau pathology is not confined to Alzheimer's disease, but it's rather quite a common final pathway of a couple of neurodegenerative diseases. So here we focus on 4 repeat tauopathies. The most common ones are progressive supranuclear palsy and corticobasal degeneration, so PSP and CBD. And depending on the pattern of tau accumulation in patients' brains, these diseases manifest in different clinical phenotypes. So we have quite a good clinical pathological matching. The most common of which are Richardson syndromes. So those are both atypical Parkinson syndromes and corticobasal syndrome. And the very interesting thing about these diseases is that we have predominant subcortical tau pathology, so in the basal ganglion brainstem, whereas Alzheimer's disease is characterized by mostly cortical tau pathology. So we have a very, very different pattern of tau distribution. And we were wondering whether our connectomic models can also predict or give information about tau spreads in these diseases, so whether there's a uniform mechanism. And so far, in these 4 repeat tauopathies, tau could only be quantified using postmortem data. And here the interesting thing is that tau does not only occur in neurons, but also in glia and astrocytes. And but now with the development of new tau PET tracers, this is data here from a colleague in Munich, we can now visualize 4 repeat tauopathy also in living patients. So this is a class of PSP patients, for example, compared to controls, and you clearly see this higher uptake in the basal ganglia. And here we then asked, can we basically use this connectomic spreading approach to recapitulate these spreading sequences that we see in PSP and corticobasal degeneration? To this end, we then again combined functional connectomics with in vivo tau PET data, and for validation also with postmortem data. So when we look at the subcortex only, so we parcelate the brain here in these colored subcortical regions and determined tau PET within these regions in a cohort of patients with 4 repeat tauopathies, and obtained connectivity between these brain regions. And here we found again that brain regions which were more closely connected to each other had a higher similarity in their tau PET uptake, again suggesting that brain regions which are connected to each other kind of share their tau levels. And when we expand this to the entire cortex, we find a very consistent pattern. So suggesting that we have again this ubiquitous phenomenon of connectivity, or the brain connection roadmap, providing information about how does tau spread across the brain, potentially in a transsynaptic manner. And when we then try to move this to patient level, and you've seen very similar slides for Alzheimer's disease, we again define the epicenter of tau pathology for a given patient. So the brain region with highest tau, and we assume that highest tau PET should be found in, besides the epicenter, in brain regions which are closely connected to that epicenter. So we expect this gradient of tau accumulation that follows the connections of the epicenter. And in these 2 plots for PSP and corticobasal syndrome, each line is a patient. And we again parcellated the brain in these 4 quartile regions. And you see that there is actually, for the subcortex, this gradient of tau PET throughout connected regions. So to remind you, Q4 is only weakly connected to the epicenter, and Q1 is really highly connected to the epicenter. And when extending this again to the cortex, then we find a very similar pattern overall. But the disadvantage of tau PET in this class of diseases is, and I mentioned this initially, is that tau does occur in neurons, but also in astrocytes and in oligodendroglia. And if you think of the spatial resolution of PET, which is 3 to 5 millimeter, then we can of course not distinguish between different cellular subsystems. So we cannot tell from where does the tau PET signal come. So to this end, we then teamed up with the neuropathologists throughout the world. So this was a large consortium headed by Günther Höglinger from Hannover. And these neuropathologists then determined tau pathology within astrocytes, neurons, and oligodendroglia throughout multiple regions of the brain. And we then here try to replicate our imaging analysis using postmortem data, and to test whether the association between connectivity and tau is actually mostly driven by neuronal tau levels, which putatively reflect this transsynaptic spreading mechanisms. So we sat down with the neuropathologists, and they told us where do they extract the brain samples. So they do this in a very standardized manner, and we reconstructed these histological atlases in 3-dimensional PET space that we can use for our imaging analysis. So we had 2 independent cohorts, all with progressive supranuclear palsy, so PSP. And then we obtained tau levels for different cells throughout these colored brain regions, and used the healthy individual sample to determine connectivity between these regions. And very in line with what we found from in vivo data, we found that brain regions with higher connectivity showed a higher covariance in their post-mortem assessed neuronal tau levels. And when we repeated this analysis for different cell types, we in fact found that this was highest for neuronal tau levels, followed by oligodendroglia and astrocytic tau. And with this, I'm almost at the end of my time, and I'm already at my last slide, to just give a very brief overview of our research in a nutshell. What are we interested in? So I think my personal interest is really in understanding disease mechanisms by combining multimodal imaging, PET imaging biomarkers with connectomics. I think knowing about brain architecture can tell us a lot about how brain diseases develop. And ultimately, we want to make clinical use of these data in order to develop like precision medicine models to predict disease progression on a patient level, which should be used for patient-tailored readouts in clinical trials, while also taking into account which factors may modulate the disease progression. So reserve, resilience, genetics, but neuroimmune mechanisms as well. And with this, I would like to thank a large group of individuals. Research is never done alone, and you can see that we used a lot of patient data, which comes from all over the world. Especially here, my colleagues in Munich did a lot of this work that I showed you, but also a lot of work was done by our colleagues in Lund in Sweden. And with this, I would like to conclude. Thank you guys very much for your attention, and I'm happy to take your questions. Thank you.
Nico, thanks so much. It was really a fantastic talk, incredibly clear even for a synthetic chemist. I really appreciate the way you took us through that. Thank you. I mean, you're just at the beginning of your academic career. I think the progress you've made in understanding sort of tau development in the brains and correlating that to disease pathology, it's really fantastic. So congratulations. Thank you. I encourage everyone, we do have a good amount of time, so please put your questions in either the Q&A box or the chat directly, and I will ask Nico. So first question from an anonymous attendee, so I'm sorry I can't see your name. Do sequence variants in tau correlate to different spreading patterns?
Yeah, this is a very good question. So we cannot really tell yet. I mean, the major difference between Alzheimer's disease and 4-repeat tauopathies is that the molecular composition of tau is very different. So in AD, Alzheimer's disease, we have 3 and 4-repeat tauopathies, or tau pathology, and the spreading pattern is very much different compared to the one of 4-repeat tauopathies. But I do not think that the molecular composition matters so much about the pattern. I think the pattern is determined by the connectomic architecture. It all depends on where does it start. So wherever I inject the pathogen, so to say, it will follow the roads that it finds. But certainly there is different spatial vulnerability for the development of certain tau pathologies, but this is not fully understood yet to my knowledge. But Michael Goddard from would probably, he's the guy who knows everything about molecular composition of tau pathology. He'd probably give a better answer to that.
Can you start to predict the start point for tau development within the brain, and therefore you have like an idea of sort of the subsequent rate of progress of tau development?
Yeah, we can try to model where does it start, and we know that it does very often start in the entorhinal cortex, so in the medial temporal lobe. And I think that this is just an age-related phenomenon. So all of us develop tau in the entorhinal cortex as we age. But I believe then when we have the second hit of developing amyloid pathology, then amyloid really drives this process of pulling tau like a vacuum cleaner out of the entorhinal cortex. But there are people with very different patterns. So for example, clinically, we see often people with very early onset AD, so who become symptomatic at the age of 50 and who progress very fast. And they show a majorly different pattern of tau pathology, so more or less in the neocortex. And it might be that their onset is different. And one hypothesis is actually that mild traumatic brain injury triggers a certain hyperphosphorylation of tau and places a seed somewhere in the brain of tau, or of abnormal tau. And then once you accumulate amyloid, then this may basically pull the trigger for the bullet, which is then tau. But this is work in progress, but we have submitted a paper actually on these different starting points, which hopefully will come out soon.
Is this the correlation to sporting injuries that are increasingly prevalent they talk about footballers and rugby players developing?
Exactly, that was one of the first publications arguing that mild traumatic brain injury triggers tau, but then the tracers also bind a bit to glial scars. So it's not entirely clear whether this is really tau or just something else where the tracer just accumulates. So we need more histopathological data of these people to really confirm that what they develop is tau.
Okay. So a question from Lisa Dunn, is tau transmission inhibited by acetylcholinesterase blockade?
Good point. No idea, to be honest. From a pathophysiological point of view, I wouldn't believe so. But to my knowledge, it hasn't been tested. So we haven't compared the rate of spreading between people who receive rivastigmine, for instance, versus who don't receive it. Could be tested. I mean, on a imaging level, what happens on a molecular level, we don't know.
Thanks, Nico. Is there any evidence that different diet or exercise behaviors correlate with different rates of spreading? And I guess the extension is that, are there any other environmental factors that affect the brain segmentation generally?
Yeah, this is an interesting point. So currently there's evidence that at least some things like physical exercise, or in general, a better vascular brain health, which is fostered by better physical exercise, increases your brain health because your brain clearance mechanisms are better. Whereas when you develop vascular damage, brain clearance breaks down, and your lymphatic system has a hard time of bringing pathogens out of the brain. So this clearly can modulate the development of primary pathology. Evidence is not, could be clearer though, I would say. But what is much clearer is that physical exercise helps you to, for example, secrete BDNF, so neurotrophic factors, which then help you maintain neuronal integrity despite having those pathological factors. So you have a higher spine density, so to say, even though you have underlying amyloid and tau pathology. So there's different levels in which resilience and resistance can come into play. So either prevention of primary pathology, but also prevention of downstream neurodegeneration.
Okay, thanks, Nico. Two more questions. First one is, are there examples of patients with tau accumulation that stalls, stops, or declines even over time? Yeah,
there has been one case report by the Harvard group. So this is a Colombian kindred study. So there's a large group of families in Colombia which carry autosomal dominant mutations for Alzheimer's disease. And they found one patient who had the brain full of amyloid because she's genetically determined to, but she didn't show much tau. And the bit of tau that she showed was just in the inferior temporal lobe. Whereas, and she was 70, I think, and at that age, she should have basically passed away already 10 years ago. She only had little cognitive impairment, so she was somehow resilient to develop tau pathology. And in this patient, they found a very interesting mutation in the ApoE gene, which they're currently following up, and how this could actually be related to limiting the spreading of tau pathology. But this is a case report, and there we see how important case reports of single patients can be to get a better understanding of pathophysiology.
And what is the time period between two different neuroimaging tests that could evidence the differences you talk about, so tau and amyloid? Of course, this will define clinical study duration and follow-up.
It depends on, always the answer is it depends.
Yeah.
So it depends on how far along the trajectory you are. So amyloid accumulates quite slowly. I'd say that we see just from by eye, you see a change usually 2-3 years roughly.
Okay.
With tau, not much is happening in the beginning, in the first 15 decades, but that's also where we do not see the patients because they do not come to the clinic because they're not impaired. But once they start to develop tau, I would say there's significant accumulation that is measurable with PET over a time period of 1.5 years, which is kind of also in line with the typical clinical trial duration that that phase 3 trials employ. But then once you are demented or when you're very severely impaired, then we get another issue of quantifying tau accumulation because the brain becomes so atrophic and perfusion becomes difficult so that less tracer is coming into the brain and has less tissue to bind to. So then we oftentimes see also a decrease of tau PET over time, at least, which doesn't make much sense from a pathophysiological point of view. So we at some point reach a sensitivity threshold, but once you're super demented, then probably there's hardly any treatment that would help you anyway. So phase or clinical trials go earlier and earlier and earlier. And then these people, I would say 1.5 years.
Okay, so just you mentioned the tracers effectively. Is there still room for development of better clinical tracers for imaging, or is this, I mean, this is a chemist question, but are we already at the state where sort of you're perfectly satisfied with the tools that are available for the PET imaging?
No, and there's actually a lot of dynamic in the field. So most of the data I showed you was using first-gen tau tracers, which are now very well established, which we know very well in terms of kinetics, binding characteristics. But the issue is they show a lot of off-target binding to, for example, MAOB, and which makes it hard to visualize anything in the subcortex. And for example, the data on 4-repeat tauopathies that I showed used the new tau tracer, which does not bind to MAOB. And here, for the first time, we can visualize the subcortical tau deposition. And there are many, I think now 3, 4, 5 different second-gen tracers, which are really promising. And which all come with a better off-target binding profile. So we get a cleaner image, because the first-gen tracers were quite noisy. The second-gen are way better, but a lot of studies refrain from switching tracer because they're longitudinal. They've done this for years, and if you switch, then your follow-up data is not comparable to your baseline anymore. But it will come more and more.
Well, okay. So next question. I'm a bit confused if increased interconnectivity helps tau propagation, why subjects with potentially increased connectivity by education exercise are protected.
This is a very smart question, which I get asked very often, so I expected this, but this means really you guys have understood what I was talking about. So there's one, we showed that increased or education and IQ are probably manifesting as resilience by having more segregated brain networks. So there might be overall increased connectivity, but it's more structured. So we have different modules, and you have to imagine also that if tau starts in one module, but this module is quite shielded from the rest, then we'll have a hard time of getting out there. So there's less intermodular connections. And actually, this is work done currently by one of my PhD students. She could show that more segregated brains actually prevent tau from spreading. So when your epicenter is more segregated, then there's not so much happening. But I agree, it's kind of a paradoxon that increased connectivity helps you maintain cognition at first, but then later is kind of the route that tau takes. But if you look at the topology that the control network has, which is more or less the superior surface of the brain, and tau typically develops in the inferior part of the brain. So there's a spatial dissociation that these brain regions are more or less maintained longer, but once tau reaches them, and then it targets hubs and spreads. So they're not guarded against maintaining brain or cognition forever.
Thanks, Nico. And then I think the last question, what is known about the cause of amyloid accumulation?
Oh yeah, tough question. That is, I would say, the million-dollar question. There's one clear cause that we can name in patients with autosomal dominantly inherited AD, because they carry basically mutated copy of the gene, which renders them prone to accumulation. So there is overproduction. So same is true in patients with Down syndrome. They have 21st chromosome 21 is there 3 times, and the APP gene is on the 21st chromosome. So there is overproduction. With sporadic cases, so who develop it just sporadically, as the name says, it's much more difficult. And one hypothesis is that it's impaired brain clearance, that clearance mechanisms cannot really keep up with the amyloid production that is ongoing. Or this is another very interesting hypothesis, that actually pathogens which may enter the brain, such as happening in periodontitis. There's actually some pathogens infiltrating the brain, that this infiltration triggers amyloid accumulation, which shields that from the surrounding. And that this may be an initial starting point, but there's probably a disbalance between production and clearance and additional factors which may bring in amyloid into the game. It's probably multifactorial, and that's why it's still undiscovered.
Okay, thanks very much, Nico. So we do have one last question, but I'm afraid in time we're going to have to stop here. But if you want to join us for the open discussion with Nico in a moment, please feel free, and I will take the questions over. I just want to say thanks again, Nico. Great lecture. We really appreciate you taking the time to join us in a really active Q&A session, which I really enjoyed. So for everybody, thank you for joining us today. We really appreciate you taking the time, and please feel free to come and join us if you want to say hello to Nico and continue the discussion. With that, goodbye everybody, and have a nice afternoon.
Bye, thank you.